Welcome to this session on the fundamentals of genetics. Today, we are going to explore a fascinating biological phenomenon known as interference. If you have ever wondered how our cells carefully manage the exchange of genetic material without making a tangled mess, you are in the right place. We will build our understanding from the ground up, starting with a simple analogy, moving through the formal biology, and finally mastering the mathematics that geneticists use every day. Let us dive in. To understand our topic, let us start with a simple analogy. Imagine you are planting trees along a narrow sidewalk. If you plant a massive oak tree, its wide roots and thick branches take up a lot of physical space. It becomes absolutely impossible to plant another large tree right next to it. You are forced to move a certain distance down the sidewalk before you can safely plant the next one. In genetics, a very similar physical spacing rule applies to our chromosomes during cell division. The physical presence of one event blocks another from happening too close by. Now that we have our tree analogy, let us look at what is actually happening inside the cell. During cell division, specifically meiosis, chromosomes pair up and exchange pieces of DNA. This swapping process is called crossing over. However, the physical machinery required to make that cut and swap is quite bulky. Because of this physical bulk, when one crossover happens, it actively prevents another crossover from happening right next to it. Geneticists call this blocking effect interference. It is a physical constraint that ensures genetic exchanges are properly spaced out along the chromosome. Building on that physical intuition, let us define this formally. In genetic terms, interference is the phenomenon where the occurrence of one crossing over event reduces the probability of a second crossing over event in an adjacent region. When homologous chromosomes align, they form chiasmata, which are the actual physical sites of genetic exchange. Because the chromosome has stiffness and the recombination machinery takes up space, a single chiasma creates a suppression zone around it. The closer two regions are, the stronger this suppression becomes, ensuring crossovers are evenly spaced rather than clustered together in one tiny spot. You might be wondering why this suppression zone matters so much to scientists. Understanding interference is absolutely critical when we are trying to map the exact locations of genes on a chromosome. If we incorrectly assume that every crossover happens completely independently of the others, our mathematical predictions for the distances between genes will be completely wrong. By measuring interference, geneticists can correct their maps. In the real world, having highly accurate gene maps is essential for breeding disease-resistant crops or tracking genetic diseases in human families. Without it, researchers might look in the completely wrong section of a chromosome. Now that we understand the biological concept, let us look at how we measure it mathematically. We start with the coefficient of coincidence, represented by the letter C. Conceptually, this formula compares how often double crossovers actually happen in reality to how often we mathematically expect them to happen if they were completely independent. The numerator is the observed number of double crossovers, which is the actual count we see in an experiment. The denominator is the expected number of double crossovers. If the observed number perfectly matches the expected number, C equals 1, meaning the crossovers are completely independent. If the observed number decreases, C decreases, meaning interference is stronger. This formula we just saw leads us to the actual calculation for interference. The symbol I represents the interference value, which tells us the exact degree to which one crossover interferes with another nearby crossover. We calculate it by subtracting the coefficient of coincidence, C, from 1. When C is 1, meaning observed equals expected, the interference becomes 0, indicating absolutely no interference. On the other hand, if we observe 0 double crossovers, C is 0, and the interference becomes 1. This is known as complete interference because the first crossover completely blocked the second one. As C decreases, interference increases, showing a stronger blocking effect. Before we move on, let us see this with a concrete example. Suppose a geneticist is studying 3 linked genes in fruit flies. Based on known map distances, she expects to see exactly 40 double crossover offspring. However, when she counts the offspring in her lab, she only observes 10. We need to calculate the coefficient of coincidence. We use our formula, dividing the observed double crossovers by the expected double crossovers. We substitute 10 into the numerator and 40 into the denominator. Dividing these gives us a final answer of 0.25. This means we only saw 25% of the double crossovers we mathematically expected. Now that we have our coefficient of coincidence, let us find the total interference for this chromosome region. We take the maximum possible interference, represented by the number 1, and subtract our calculated coefficient, which is 0.25. This simple subtraction gives us a final interference value of 0.75. Physically, this final answer means that 75% of the expected double crossovers were suppressed or blocked by the occurrence of the first crossover in that region. This proves that a strong physical blocking effect is taking place on this specific chromosome. Let us look at a slightly more complex scenario, where we must calculate the expected double crossovers ourselves. In a 3-point test cross, the distance between gene A and B is 10 map units, and between B and C is 20 map units. Out of 1,000 total offspring, the researcher observes only 5 double crossovers. First, we convert map units to probabilities. 10 becomes 0.10, and 20 becomes 0.20. We multiply these independent probabilities together to find the overall probability of a double crossover, which is 0.02. Finally, we multiply this by the 1,000 offspring to get our expected number of 20 double crossovers. Continuing with our test cross, we now have our expected double crossovers, which is 20, and our observed double crossovers, which is 5. We divide the observed by the expected to find the coefficient of coincidence. 5 divided by 20 gives us 0.25. Just like before, this indicates only a quarter of the expected crossovers took place. To find the interference, we subtract this coefficient from 1. 1 minus 0.25 gives us a final interference value of 0.75. Once again, the physical presence of one crossover reduced the occurrence of a second nearby crossover by 75%. Now that we have calculated interference mathematically, let us visualize exactly what is happening. On screen, you see a diagram of 2 homologous chromosomes, one in red and one in blue. They are crossing over at a specific point, forming an X shape known as a chiasma. Notice the shaded area surrounding this intersection. This is the interference zone, or suppressed region. Because the physical machinery making this genetic swap is bulky and the chromosome itself has stiffness, it creates a physical constraint. As the label explains, this constraint actively prevents a second crossover from occurring within this shaded area. This visual perfectly captures why our mathematical interference values exist. We have covered a lot of ground today, so let us summarize the most important ideas. First, Interference is a vital biological safeguard that prevents multiple genetic crossovers from happening too close to one another on a chromosome. Second, this happens because the physical machinery required for crossing over is bulky and creates a suppression zone. Third, we can measure this mathematically using the coefficient of coincidence and then calculate the exact interference value. An interference of 1 means complete suppression. Finally, understanding and calculating this concept is the absolute foundation for creating highly accurate DNA maps, which are essential for modern genetic research and medicine. You now have a solid grasp of genetic interference.